In this section, we will be discussing some important ideas about very unique form of connective tissue, which is the blood. But certainly, it would be much more beneficial for all of us to make a little bit of introduction into cardiovascular system, which handles the blood, so that we become simultaneously knowledgeable and to have some basic understanding of how the cardiovascular system is arranged, and for that reason, how it handles the connective tissue, which is in a liquid form, that is blood. Most likely that it would be a bit too much to fit everything into a single recording, so please pay attention that I will label them properly. So it would be part 1 and part 2, and feel free to check both of them. So this diagram is ideal to give us at least a basic breakdown of how the cardiovascular system is organized. Certainly in the center of the screen, we can see the heart, the principal pump, which keeps receiving blood coming at one end and then pumping the blood out at the opposite side end. For that reason, within the heart, we will be discussing later on two smaller chambers that are receiving the blood. They're called the atria. And atria move blood directly into two larger chambers known as the ventricles, which upon their contraction will pump out into two connected, but we can say for learning purposes, for educational purposes, we can say two different components of the cardiovascular system. We're talking about pulmonary and systemic circulation. You can see also that the difference in color on this diagram, blue and red, essentially tells us a little bit of a story behind these two colors. Blood, which is returning to heart, is known to be deoxygenated. It delivered the oxygen, it delivered the nutrients, and now has to come back for the purpose of reoxygenation. For that reason, we are starting usually the presentation of cardiovascular system within the right atrium, a smaller chamber which receives venous deoxygenated blood from two large blood vessels. One of them is called the superior vena cava. The other one is known as the inferior vena cava. Word cava means hollow. There's a third blood vessel also, which enters the right atrium, and it is a coronary sinus, which collected venous blood from the heart itself. But relative to the size of this diagram, it's a bit small, and for that reason, it's not included in this illustration. A moment, blood is within the right atrium. Atrial short-lasting contraction will make sure that the blood is pushed down into right-sided ventricle, from which it will be directed into a single blood vessel known as the pulmonary trunk. This is what we can say the beginning of the pulmonary circulation. Deoxygenated blood goes from a right atrium, right ventricle into pulmonary trunk, and then via left and right pulmonary arteries, it's going to go into lungs for the purpose of oxygenation. Once the blood circulates through capillaries of lungs, both left and right, it'll start its journey backwards and through the pulmonary veins, illustrated in red color. Oxygenated blood will find its way back into the heart, but this time into left atrium. So this short-lasting journey between blood entering a right atrium and returning to heart, but into left side atrium, is what we can say defines the pulmonary circulation. Speaking of systemic circulation, once the oxygenated blood enters the left atrium, atrial contraction pushes it down into left ventricle, which then contracts on its own, sending the blood into single largest blood vessel in the human body, not the longest, just the largest because of its caliber, and it is aorta. Aorta, through its branches, will make sure that oxygenated blood is properly distributed to every single part of the body, whether it's head and tail and neck, whether it's into upper or lower limbs or into body cavities, into thoracic, abdominal, or pelvic space. And as such, of course, arterial oxygenated blood is distributed evenly throughout the body. But perhaps we have to set one simple and easy to follow rule. Obviously, word artery and word vein is going to be used multiple times. So what defines blood vessel to be called an artery? It has to bring blood away from the heart. So for that reason, aorta is defined as the artery. Pulmonary trunk is the artery. Pulmonary arteries are, of course, given the same name because all of them are taking blood away from the heart. Blood vessels bring blood back to the heart, whether they are known as the left or right pulmonary veins or superior or inferior vena cava. Obviously, they bring blood back into the heart's chambers, and that is what defines blood vessel to be a vein, to bring blood closer back to the heart. Although these are major blood vessels, both arteries and veins, when we pass the level where we can macroscopically identify them, arteries become smaller after dividing multiple times, giving rise to arterioles. And then arterioles, as they also keep dividing, are finally there to produce the finest and most impressive part of the cardiovascular system, the capillaries. Because the walls of arteries, venules, veins, arterioles are still too thick, there would be no transport through the wall of artery or arteriole, venule or vein. It's the capillary bed which allows direct interaction between blood and other tissues. Blood brings oxygen and nutrients, but also at the capillary level, blood collects carbon dioxide and other waste products of our metabolism. So if that defines the system, which we can say that it goes away from the heart through arteries that gradually become smaller and divide and become arterioles. And then arterioles give rise to capillaries. Capillaries at one point start collecting more blood and they form venule. And union of multiple venules finally is going to result in appearance of peripheral, macroscopically visible vessels that we call veins. Same rule applies to both pulmonary as well as systemic circulation. So if we try to compare artery to vein, we can see that their walls are built up in a very comparable form. Upper part of the diagram illustrates one artery and one vein, and we can see that both of them would have internal coat, tunica interna, made by endothelial cells. Veins would have endothelium occasionally thrown into folds that are called valves to ensure that venous blood has unidirectional flow so that the blood doesn't regurgitate backwards as it could be under influence of gravity, for example, for lower limbs. Then the middle coat is called the tunica media. It is made by smooth muscle fiber tissue. Clearly, arteries have a little bit more of the muscle tissue within the wall, as opposed to veins that have only sporadic presence of smooth muscle fibers. The difference comes right away from tissue resistance. Artery pushes blood further away from the heart and is experiencing gradually higher resistance by tissue. So contraction of smooth muscle fiber will be there to increase the tone of an artery and force blood to keep moving away from the heart. Veins do not face the same tissue resistance. As a matter of fact, they are experiencing less and less of a resistance because venules start from a level of tissue and then they progressively become veins that are facing practically no resistance any longer. And for that reason, the role of smooth muscle fibers is not deemed crucial for veins. Finally, the outermost coat or layer, the tunica externa or adventitia, quite slippery coat covering both arteries and veins, preventing them from being pinched between, let's say, two muscles while they're producing contraction, or between a muscle and a bone, or any other tissues that eventually would start compromising the flow of blood, which should not ever happen. Blood must keep flowing before it accidentally would trigger the process of coagulation, blood clotting. For that reason, blood keeps moving continuously throughout our lives. Now, in a contrast to both artery and vein, we can see at the bottom of the screen what one typical capillary looks like. And it's not going to take long for us to conclude that it's essentially made of a single layer of endothelial cells being attached to the basement membrane, as it would be expected. It's going to be too much to have flattened epithelial cells practically sticking so firmly to each other to maintain the integrity of the wall. So it's the basement membrane, which is an important part of capillary. This is cross-section through one artery, and we can see here those three layers. We can see elastic, lamina, and endothelium. We can see here the muscle layer. We can see external coat, external layer, tunica externa. And here at the lower part of the screen, we can see what's the size of capillary. So practically it's wide enough to let just one by one red blood cells passing through. If I give you an idea that a red blood cell on average measures 8 micrometers, basically capillary might be just a little bit wider than this in order to ensure that red blood cells can pass easily. Of course, contraction and relaxation of the heart is going to be deemed most important for proper start of this system, although heart, through its contraction, won't be able to keep moving blood on its own. That's why smooth muscle fibers, particularly in the walls of arteries, are going to be of major role for the well-being of the cardiovascular system. However, capillaries do not always behave the same. One can say that they are really there to integrate themselves between the blood and any other living tissues. But as we know, the tissue has a variable demand for oxygen, variable demand for nutrients, depending what current situation we're experiencing. So, for example, when we have a need to properly feed, let's say, group of muscles, of course, local area blood vessels are going to do their best to supply muscles with, we can say, almost unlimited amount of blood that would be required for their contraction. And diagram on the left side illustrates that from this blood vessel, named as the met arteriole, obviously being the main thoroughfare channel between arteriole and venule, basically there are some smooth muscle fibers acting as capillaries. When they're fully relaxed, blood, of course, starts filling the rest of the capillary bed. On the right-hand side, muscles not active, they do not require the same amount of blood as they needed during their maximal contractions. In that case, those smooth muscle fibers are going to be acting as sphincters. They would contract, and they would prevent massive movement of blood into capillaries that currently do not support the metabolic function of that part of the body. So the blood will be allowed to pass through met arteriole, and perhaps priority in delivering more blood will be allocated somewhere else. As we know, for example, after a major meal, we would have a need for more blood being shifted into digestive tract in order to support proper digestion. And it is never recommended to go and immediately start practicing some kind of heavy physical exercise by body using our sheer muscle strength, because locomotor system will take blood away from a digestive system, and they become at that point slightly incompatible. So this is just to illustrate why do we have to alter blood flow through capillary beds, basically to match the current metabolic requirements. The word capillary came quite a few times over the last few minutes of this presentation, and I just want to mention that not all capillaries are built exact the same. Practically, if you just go into the basic configuration, you can say, yes, it's always endothelial cells and the basement membrane, but you can see that we have really three different types of capillaries, which we would encounter within the human body. The most common type is known as the continuous capillary. And what is the continuity? Basically, presence of very small gaps that exist between individual endothelial cells is determining the outcome as which substances eventually could pass between two endothelial cells, both from capillary into tissue direction or tissue back to capillary. So we can say that this practically sets the norm or standard. But then we have some capillaries that are titled fenestrated capillaries. Fenestra means window, so we have lots of different openings, fenestrations or pores that are going to be a greater size, and therefore some molecules that are larger than the gap between endothelial cells will be able to go through fenestration. And this is typical for kidneys, where we expect kidney capillaries to allow us to filtrate what appears to be almost a volume of 200 liters per day. And finally, we have this type of capillary called the sinusoid capillary, where size of the openings between endothelial cells or within the endothelial cells is so huge that essentially even the largest molecules and a quite copious amount of fluid can come out. Typical good example of where sinusoid capillaries are found is within the liver, within spleen, where it's not really aimed to just allow massive movement of fluid from cardiovascular system into tissue, but rather it significantly slows down the movement of blood, ensuring that both liver and spleen would have sufficient time to properly process the blood which arrived into these organs and to complete their metabolic role. So this is just to clarify the idea of veins having multiple folds of endothelium or tunica interna forming valves. Arteries don't have them, only veins. So if the blood would be reversed and try to go backwards from top to the bottom of this diagram, of course, the blood will start filling up the pockets between the walls and the valvular leaflet, forcing the valve to close, and no further movement of blood downward would be allowed, especially in a healthy adult. It is going to be very important, but as we know, sometimes failure of these valves would result in the formation of quite enlarged varicose veins, lower limbs in particular. So we can say that this practically completes the introductory part to cardiovascular system. We're going to come up to this same slideshow when we do cardiovascular system full anatomy and physiology. But for now, I think that we can go to a different slideshow and start introducing the tissue that we already introduced at the beginning of this recording. It is the blood. And let me just close this slideshow and open the next one, which brings us to the main topic of this recording, the blood. If we recall what connective tissues are made of, basically we're going to say cells and matrix. Matrix made by fibers and ground substance. In this case, obviously cells are going to be quite directly visible, whether they're red blood cells or white blood cells. But when it comes to matrix, we're going to have no fibers. And of course, the ground substance is going to be practically very simple to understand. water. If we're to understand what the composition of the blood is, we have to perhaps think about what is this vial showing to us. If a sample of the blood is filling up the testing tube and it's put into a centrifugal machine, basically there will be a separation of foreign blood elements from liquid component. Foreign blood elements, which are heavier, whether they're red blood cells or white blood cells or platelets, are going to start separating from liquid within which they're suspended. And as a result of their further separation, basically we're going to have at the bottom of the vial about 45% of volume, full blood, will be red blood cells. Then we're going to have fairly thin layer, which is called the buffy coat with a very light color. It is composed of white blood cells and very small cellular particles that are known as platelets. So we can say that their volume is quite negligible, and therefore we can allocate about 55% of full blood to make plasma, which is water, and a variety of different suspended particles that we're going to find out in the lower part of the screen. So the whole blood practically makes about 8% of ideal body weight for some average size adult human. We know that it would translate to about 5 liters of circulating blood per average person. And of course, we need to break it down, and we already came with numbers that the blood plasma is 55, and then the rest of the formed elements will make 45% of the volume. Now, one thing that I also want to add, I kind of intentionally tried to stay away from terms red blood cells, white blood cells, and then platelets. Red blood cells are called erithrocytes due to their red color. White blood cells are called leukocytes. Leuko means pale or white. And thrombocytes, although the suffix "cytes" would point out that there are actually cells, they're really not cells. They are formed blood elements because they come as particles that have been kind of shedded from the surface of a large cell. So platelets truly are not cells. They're thrombocytes, but they're fragments of a cell which produce them. Liquid part, which we call the plasma, and accounts for about 55% of the volume, we need to understand its chemical composition is of 55%, 91.5% is just plain water. However, the second largest ingredient, which we have to add, are proteins. And proteins that are part of a plasma are very important because you can see further breakdown of all proteins. Albumins appear to be the most abundant, globulins in the middle, fibrinogen, practically the least frequently encountered type of protein, and other proteins that account for just 1%. All of them are synthesized within the liver. All of them are released into bloodstream from liver, and they do have somewhat different function. Some of the albumins and globulins are going to be interesting because they will assist in the transportation of molecules through the cardiovascular system. We didn't say it, but it's always easy to understand what is the principle or what are the principal roles of the cardiovascular system. First one is transportation. The liquidity of blood definitely ensures that liquid can reach even the most distant parts of the body and deliver whatever is needed, whether it's oxygen or nutrients or both. Then the blood will have its regulatory role. Regulatory role is to maintain certain homeostatic values, one of them being our body temperature. So when we have increased body temperature, we know that we're going to see a little bit of extra redness on our face or any other visible parts, because there would be an increased blood flow towards peripheral parts of the body from which the heat can easily evaporate. And opposite will be seen when the body temperature keeps getting lower than our ideal homeostatic value. That'll result in shrinking and contraction of major peripheral blood vessels, trying to conserve the heat and keep it more within the core parts of the body rather than in the most peripheral parts. That's the reason for which our hands get quickly cold during the winter if they're not protected, our feet, or any other end parts of the body, such as the nose or ears in particular, which could be subject to some kind of changes, especially when exposed to severely cold weather. Third and final role is protection. And although the word protection appears to be very wide, we know that we're going to have some components of blood being really like irreplaceable parts of our immune system, which will help with overall maintenance of health. And it is going to be mostly coming from a population of leukocytes or white blood cells. So what are these proteins actually good for? Albumins are going to have mostly the role for transporting larger substances like lipids, because the combination of protein and lipid could result in easier handling of otherwise hydrophobic lipid molecules through the cardiovascular system. When they become attached to a carrier molecule of protein, basically their hydrophobic nature immediately diminishes, and the rest of the body is able to handle it properly. Albumins, due to their high 54% presence in liquid part of the blood, will also have a very strong attraction for molecules of water to come and stay next to molecules of albumins, because they are hydrophilic. That prevents that, as the blood runs through capillaries, eventually too much of a fluid loss would be encountered, tissue retaining the water, drying out blood. Globulins would join albumins, but primarily they become part of our immune system, and that's why sometimes people simply say we're calling them immunoglobulins. The fibrinogen, present as just 7%, is an interesting protein which we need for one major eventuality that will affect all of us at least multiple times through the life. We sustain a simple, small cut damage done to finger, to toe, or any other part, which we frequently expose to a variety of different injuries, especially when we were younger, scratching elbows or knees or something like that. So what's the common site that we see on the site of an injury? Blood, because integrity of skin, integrity of some blood vessels have been disrupted by this traumatizing event. As a result of blood's ability to start fixing the damage right away, one of the most important things to prevent uncontrolled loss of blood is going to be seen in the form of blood coagulation or blood clotting. And as the blood starts plugging the defect, preventing more profound blood losses, it's the fibrinogen is predecessor molecule which needs to be activated. Fibrinogen becomes fibrin, and then it starts trapping the platelets and forms a blood coagulum, which closes the gap, and the bleeding at that point stops. Other than water and proteins, we know that there are other solids that are also found within plasma. There are electrolytes, which are practically matching the composition of the body. Blood cannot have excessively more or excessively less potassium or sodium. It has to be something that's compatible with both intra and extracellular fluid compartments. It carries various amounts of nutrients after we just completed meal, of course, the amount of nutrients will go up, but in the process of fasting, avoiding any kind of food caloric intake for 24 hours, volume of nutrients within the blood is going to go significantly down. Gases, of course, oxygen, carbon dioxide, regulatory substances, whether they are hormones produced by glands with endocrine secretion, they're using blood as a transportation mechanism, or maybe we can just say sometimes vitamins also regulate certain functions. And finally, the waste products, which blood through the capillary bed collected from tissues, completing their metabolic cycles, and is carrying them away to the point where these waste products of our metabolism are going to be eliminated, which carbon dioxide, certainly we can easily exhale, but some other byproducts of our metabolism we use to have urinary or digestive system that will excrete them and prevent any kind of significant accumulation within our body. Okay, if this explains what the plasma looks like, let's talk a little bit about formed blood elements, which compose 45% of full blood volume. We know that there are three basic types, and we're talking about platelets, therefore fragments of cell. Thrombocytes would be their more scientific name. Then leukocytes, or white blood cells, where they're present within 5 to 10,000 white blood cells per microliter of blood. Microliter, if you just want to get a little bit of idea, is something that you imagine as tiny cube made by 1 by 1 by 1 millimeter sides, and you fill it up with blood. That is the volume of 1 microliter. So 1 cubic millimeter is what contains 5 to 10,000 white blood cells. And that same 1 cubic millimeter of blood would be good enough to have additional 4.5 to 5.5 millions of red blood cells, or erythrocytes. I didn't mention, but total number of platelets in adult average healthy person should fall within this range between 150 to 400,000 thrombocytes per microliter of blood. And as you can see, we have a need to go one more column to the right, because obviously, although all the red blood cells, regardless of their massively abundant presence, are practically the same, amongst the class of white blood cells, we have 5 different subclasses. And as you can see here with their percentages, the most common type of white blood cell is known as the neutrophil. Then we're going to have between 20 to 25 lymphocytes, 3 to 8% of monocytes, 2 to 4% of eosinophils, and only between 0.5 to 1% of basophils. You realize that there are 3 of them which end up with a suffix phils: neutrophils, eosinophils, basophils. They're also called the granular leukocytes because within their cytoplasm, if you come much closer to the screen, you can see some kind of interesting granulation, although these diagrams are really small. Basically, these granulations is what puts them in the class of granular white blood cells. Eosinophils and basophils were given that name after basically they have been dyed by acidic or alkaline substances. Their granulation accepted either acidic or alkaline dye, hence eosinophils, acidic dye, basophils, alkaline dye. In neutrophils, neutral, we're obviously accepting both acidic as well as slightly alkaline dye, and their granules have changed the color. Other 2 types, the lymphocytes and monocytes, are agranular white blood cells. They do not show presence of granules, and also you can see that their nuclei, appear to have a nice rounded form, where the granular white blood cells have the nuclei in a somewhat segmented form, which I believe can be seen much better and more direct on some of the diagrams that are to follow. Here is a beautiful picture of sample of blood, which immediately shows multiple red blood cells shaped like biconcave discs, because although they are cells, what really is their major characteristic is that they do not have nucleus. Actually, it's better to say that they used to have it once upon a time, but the nucleus has been eliminated from the cell, and that's why it allowed the red blood cells to have somewhat flattened form in the form of, as we said, biconcave disc, almost like a donut. White blood cells do not appear to be flattened. They're nice and rounded, and in between them we have those irregularly shaped parts, the platelets or thrombocytes. Magnification used 3,000 times so we can see it as directly as this picture illustrates. So here is a little bit more about blood cells, or better to say red blood cells, white blood cells, and platelets. Where do they come from? We know that if we are asked this question, where all the blood comes from, most of us would say right away that it comes from bone marrow. It's a common knowledge. But why and how can we explain such a variety, 5 different subtypes of leukocytes? Basically, this diagram will answer it to us. We would have in our past the pluripotent stem cells, which moved and migrated into bone marrow quite some time ago during our embryonic and fetal development. And those pluripotent stem cells are responsible to build both 2 different classes, whether we call them myeloid stem cells or lymphoid stem cells. You're going to see in the next couple of minutes what the difference is. So myeloid stem cells, of course, they have to further differentiate, and they would become progenitor cells, which later become precursor cells, and then precursor cells finally give us either formed cells or formed blood elements of circulating blood. Some of these cells would also reside within a tissue, and as such, they would become a very important and interesting addition to our lymphatic and immune system. So if we follow the path of pluripotent stem cell and its further, let's say, changes leading to appearance of myeloid stem cell, this one can be responsible, and we see here that it produces red blood cells, as well as platelets, as well as eosinophilic, basophilic, and neutrophilic cells, including monoblasts. So let's see, myeloid stem cell is producing pro-erythroblast, which becomes immature cell called the reticulocyte, which has a role to eject its nucleus before it finally becomes mature red blood cell or erythrocyte. Myeloid stem cells also could produce cells that are called megakaryoblast. Karyon is a nucleus in Greek. So megakaryon, large nucleus, blast, identifying cell that is metabolically very active, and it will become megakaryocyte once its metabolic activity is slowed down. And perhaps you can see here some kind of cloudy discoloration around these cells. Essentially, these are cellular fragments that the megakaryocyte releases into plasma, and these fragments are platelets or thrombocytes. As we already know that there are 3 different types of granular white blood cells or granular leukocytes, here they are: eosinophilic, basophilic, and neutrophilic. Their nuclei are segmented, but because of presence of granulars, we call them granular leukocytes. Here is a little conflicting moment, which definitely needs to be explained. Okay, if myeloid cell is producing neutrophils, how come that it is also producing those monoblast cells generating monocytes? So this is a granular form of white blood cell, monocyte. And as we see, the monocyte is not necessarily linked to stay only as part of the circulating blood. Basically, its further evolution will result in the appearance of cells that are called the macrophage cells, macro, large. To phagein means to consume, to eat. So macrophage cells present, again, practically throughout the body are those ones that will help us get rid of any kind of bacterial contamination, live microorganisms, or even fragments of old and dying cells. They're going to be consumed by macrophage cells. So that is myeloid stem cell class and the products that are coming out of it. The pluripotent stem cell might take another turn and eventually give rise to lymphoid stem cell. Lymphoid stem cell produces just one type of white blood cells. They're called lymphocytes, but there are two different types, T lymphocytes and B lymphocytes. T lymphocytes and B lymphocytes are going to be impressively important component of our immune system, whereas the B lymphocytes in particular could become plasma cells, and plasma cells will be able to quickly release wide, massive amount of antibodies, the proteins that will, we can say, momentarily start dealing with any kind of intrusion into our body, especially if it is done by microorganisms that we have already encountered. So I'm talking about immunity that we have gained quite some time ago when we were children, preschooler, or during elementary school, but immunity that we have acquired at that time practically is becoming permanent, and for as long as the body is properly functioning, basically that kind of experience from our early days is not going to be forgotten. About red blood cells, we have to say one thing. Here is the measurement. I mentioned it earlier, 8 micrometers across, barely squeezing through about same size lumen of capillaries. Biconcave disc, no nucleus, and that's why it has a very unique shape. They won't last forever. Their average span is between 3 to 4 months, 90 to 120 days, after which their membranes become a little bit more rigid, preventing them to go easily through very tight capillaries, and at one point, basically attempt to squeeze the red blood cell into a very tight, very narrow capillary will result in its membrane being bursted, practically bringing the end of life to a red blood cell. And of course, they will be quickly replenished by new ones, so it is not our concern. And more massive form of elimination of older and worn out and frail red blood cells is happening within the spleen, and to some extent also within the liver, where the sinusoid capillaries are those ones that are testing mostly the integrity of red blood cells. Inside a red blood cell, since it doesn't have nucleus, we know it came to the end of its life cycle. It won't be able to multiply, so it's the G0 phase, is a very interesting molecule. It's the molecule of hemoglobin. Obviously, it is some form of protein, illustrated in multiple shades of blue, onto which we have up to four heme containing additional molecules. So that's why it's hemoglobin, beta polypeptide chains, and up to 4 different heme-containing molecules to make it hemoglobin. We don't need to worry about exact chemical structure of heme, but what is important to note is that it contains ionic iron with 2 positive charges. So that is in the core part. And we know that iron is one of the most precious form of minerals that we do not have in a vast abundance throughout the body. So it would be imperative that upon the breakdown of a red blood cell and further breakdown of molecule of hemoglobin, to do something that will preserve the iron and eventually bring it back into bone marrow, where it's going to be recycled and rebuilt into newly synthesized molecules of hemoglobin carried by newly produced red blood cells. This cycle is quite predictable and could be quite nicely summed up in this diagram. So with idea about 120 days being a maximal lifespan for red blood cells, they would be entering different parts of the body where, as I said, the elasticity of their membrane will be tested. And if not elastic enough, it is going to burst under pressure, where a red blood cell is now forcefully squeezed into, let's say, 7 micrometer capillary. Size of a red blood cell is 8 micrometers. You try to squeeze it into 1 micrometer smaller. If elastic, a red blood cell will tolerate. If non-elastic, it'll burst. So the death of red blood cells is definitely happening. And although the process of phagocytosis might be activated anywhere, specifically most of it within the spleen, liver, as well as bone marrow, we know that product coming out of red blood cell, the hemoglobin, is going to be immediately split into globin and hem containing part of the molecule. Further breakdown of globin as its long chain of protein will yield individual amino acids. They won't be used for metabolic purposes of synthesizing energy, and individual amino acids are reused for protein synthesis anywhere in the body, so we're just freely recycling them. With a heme part of the molecule, it is important to try to extract iron as quickly as possible. So iron will bond to protein also, but transport molecule, which is called the transferrin, which will get into bloodstream and bring iron back into liver. The rest of the heme will be converted into biliverdin or bilirubin. Basically, they're byproducts of hemoglobin breakdown, and they would also easily be carried through bloodstream to reach the liver. Liver will eliminate bilirubin through production and secretion of bile, and we know the bile will reach the small intestine. Within the small intestine, the bilirubin will be further converted or further broken down to urobilinogen, which becomes absorbed back into bloodstream, and the bloodstream takes it down to kidneys, where kidneys convert it to urobilin and excreted out with urine. Practically, this distinct yellowish discoloration of urine is directly related to concentration of urobilin within it. Darker the urine, more urobilin, and when the urine is dilute, as we consumed a significantly larger volume of water, or we ate multiple slices of watermelon, basically the urine will be with less and less yellowish pigmentation, sometimes as clear as the water itself. The remaining urobilinogen that stays within the gut becomes converted to stercobilin in the large intestine, and as such, basically is not further absorbed. It mixes up with rest of materials that we have not absorbed, and it's ready for elimination, so practically turning into fecal material. This, again, very distinct brownish discoloration of human feces is due to presence of stercobilin, and of course, that explains how do we excrete part of the molecule which can be easily resynthesized within a body. But we came to this point where iron has been bonded to a molecule of transferrin, and from a tissue where red blood cell was destroyed, ferritin brings iron back into liver. Liver will kind of release iron from transferrin, bind it to another carrier protein, ferritin, but shortly after will reattach the same ion back to transferrin molecule that is going to carry it from liver through the bloodstream into bone tissue. Within a bone tissue, part of it, of course, is going to be spongy bone tissue, most typically filled with red bone marrow. So in a red bone marrow, basically iron will be resynthesized with the globin with the help of vitamin B12, and this is a hormone, erythropoietin, basically, which comes from kidneys, that stimulates and supports erythropoiesis. That is the process which generates new red blood cells. So erythropoiesis, once it has produced a new class of red blood cells that are mature and ready to go, will be released into circulation, and after about 120 days maximum, it is going to affect them in the same form as what this diagram already illustrated. Another example of a negative feedback loop, and it explains the role of hormone, erythropoietin, and complete numbers of red blood cells within the circulatory system. So whatever is a stimulus, it could be a person who sustained a massive external hemorrhage due to accident, or any other condition that eventually would result in less than needed oxygen being delivered to tissues, but in particular kidneys, because they would be the organ that responds to this. So if they're not properly perfused with oxygenated blood, kidneys are able to detect the lower than normal oxygen content, and as their direct response, they would increase presence of hormone, erythropoietin, which is secreted into the blood. So extra erythropoietin reaches what we see as a controlling center. Controlling center is red bone marrow, containing cells that are known as the pro-erythroblasts. They're on a standby. They do not necessarily go and produce more red blood cells than what's just a maintenance daily routine. But we're talking here about some kind of disruption from a homeostasis, something that requires a little bit more significant involvement of red bone marrow. So under the influence of erythropoietin, more proerythroblasts mature. They become reticulocytes. Reticulocytes are red blood cells that still have nucleus. They would be entering the circulation, and of course, they would be maturing, shedding out their nucleus, turning themselves into red blood cells that are now increasing the numbers of total circulating red blood cells. Keep in mind, between 4.5 to 5.5 millions per microliter of blood. Of course, more red blood cells means more hemoglobin containing structures within the blood, more oxygen to be bonded, more oxygen to be delivered to tissues. And as such, of course, tissues becoming properly and fully perfused and given sufficient amount of oxygen practically brings the condition back to controlled level, to homeostasis. And at that point, the negative control loop will interrupt this cycle, because we do not need to overflood the system to make it, instead of 5 millions, 7 millions red blood cells. That'll add to overall viscosity of blood. Blood is 5 times approximately more viscous than the water. And we know that water will have less resistance passing through a hollow tube. Blood, due to its viscosity, will actually keep passing through tubular space, artery or vein, but because of its pretty much cellular content, it is going to experience also some friction, some resistance. So that's why it's imperative not to allow blood to become too thin or too thick, because in both ways, it will start changing those natural settings, the way that the body is best when operated at a homeostatic level. Here is a photograph of 5 different types of white blood cells. So you can see the granular, eosinophil, basophil, and neutrophil, and agranular and somewhat different shaped lymphocytes and monocytes. We're going to stop here, because I think if we start moving into direction of what is going on, what is one of the qualities of white blood cells, or how do we actually control the damage when the wall of blood vessel is eventually leaking some blood, I think that's going to be better for second part of this video. So at this point, we're going to just stop, post this recording, and come back as part 2, which will discuss the rest of this presentation, homeostasis of blood type, blood clotting, etc. Thank you for watching it, and hope to see you soon.